Hi, ladies and gentlemen. Welcome to the first lecture for the biotechnology unit. This is Chapter 20. We're going to cover basically chapters 20.1 and then a little bit of 20.2 in this video, just to give you an idea of how basic biotechnology works when we get to when we're talking about recombinant DNA, and then also when it comes to gel electrophoresis, which we're going to do on Friday. So I want to give you kind of a little bit of an overview before we get to that point. So we'll go over that, and then I'll give you the link to run through the lab as well, just so you get an idea of what we're going to be doing on Friday. So here's Chapter 20. Alright, so basically what we've got going on in biotechnology is the really cool part of basically research in biology. We've got work with DNA, we've got work with genetics, and we're trying to, see how we can get different genes expressed in different organisms and maybe in different spots to see how that affects what's going on biologically in what we're studying. So we're going to first start off by talking about recombinant DNA, which is the basics for the chapter here, and then we'll get into a little bit on electrophoresis towards the end. So basically what we've got going on here is when we're talking about recombinant DNA, recombinant DNA is basically DNA that is from two different sources. So what you'd have most likely here is DNA from bacteria and then DNA from a gene of interest. And that can be from a very wide variety of organisms in which you'd want one particular gene from to study and look at. So recombinant DNA is the making of a particular piece of DNA that combines most of the time bacteria and then whatever gene you want to study. And when it says in vitro, that means you're doing that in the lab. So most of the time this is done in the lab. So what does this help us with? Well, it can help us genetically engineer certain organisms to do what we want them to do. For instance, crops, we can genetically engineer them to, take on harsh winters more easily, or more easily than other plants would. And this kind of engineering and research has really affected the field of biotechnology quite a bit. There's so much research in the biomedical field in this type of field that, there's a lot of room to study, a lot of room to grow in this topic here. So, all right, moving on to chapter 20.1, we're going to look at DNA cloning. And when scientists want to look really at a gene and see what it does or how it can be expressed in different organisms, they use a process called DNA cloning to get that done. And DNA cloning can happen with the use of recombinant DNA. So we're going to look at that again. What you first have to start out with when you're creating a piece of recombinant DNA is you have to start out with a plasmid. And a plasmid is a nice circular piece of DNA found in bacteria. Eukaryotic cells really don't have this kind of DNA, so it's very particular to bacteria and prokaryotes. Those are the only organisms that will actually carry these around. But they're actually very good in carrying genes of interest around, and in doing that, you can create these DNA clones very easily. So what really is happening here is that foreign DNA from whatever gene you're looking at, you can even say, for example, let's look at the insulin gene. We can take the gene for making the protein insulin, and we could insert that into a plasmid. Okay? So by inserting the foreign DNA into a plasmid, we're combining two pieces of DNA, and that in itself is a recombinant plasmid then. So now all we got to do is put this into a bacterial cell, let that bacterial cell grow and reproduce, and now we've got many different bacterial cells with multiple copies of the gene you wanted. So you really didn't have to go into many eukaryotes and extract a lot of insulin genes. You just really had to get one, and then put that into a bacterial cell, allow it to get copied, and then you've got as many as you pretty much like. Here's some other examples of it, and here's a great diagram showing what we've got here. So here's our plasmid inside of the bacterial cell. Again, it's additional DNA in the circular form. And what we've got is we're going to basically cut open a plasmid. I'll show you how to do that here in a second. And we're going to insert a foreign gene into the plasmid. As soon as we get that done, we'll insert it back into the bacteria, and now the bacteria will grow and reproduce until until we basically have a colony of bacteria that have that particular gene. Now we could either do some research on the gene and extract the gene for purposes we want. Maybe we're going to, insert it into plants and make them resistant to pests, or maybe we could allow the gene to be translated into a protein, and then we could harvest the protein. And for instance, we've got human growth hormone here. We could have the gene for human growth hormone translated into a protein, then we just harvest that product. So there's a lot of different ways you could go with this, but this is really the basic first step in cloning a gene and utilizing it. So how do we cut open a plasmid and insert a foreign piece of DNA in there? Well, we use a restriction enzyme, and a restriction enzyme cuts DNA just like scissors do. And actually, I like to think of them as crazy scissors. My mother, when I was a kid, used to do like crafts, and she would have zigzag scissors and wavy scissors and like all these weird kind of scissors. And restriction enzymes really function in that kind of a way. They don't cut DNA the same every time. There are different types of restriction enzymes, and they all cut DNA in a particular way or with a particular pattern. And so picking a particular restriction enzyme is important in figuring out what spots are going to cut it at. So again, restriction enzymes will cut DNA at specific spots or restriction sites, and the pieces of DNA you make are now restriction fragments. So the pieces here of DNA are restriction fragments. After the restriction enzyme finally cuts this DNA, it cuts it in a staggered way, leaving sticky ends. And these sticky ends assist in bonding to the foreign DNA. So it's not just a clean break, it's more of a kind of ledge for the DNA to land on. Okay, so here we'll give you kind of an idea of what that looks like here in a minute. Okay, so we were right here, I believe. Sorry about that. So again, the sticky ends, that's what we're going to be creating. In the end, as soon as we've got our plasmid that's been cut open and our gene of interest that's been inserted, the enzyme ligase, just like from DNA replication, is going to come back and is going to covalently bond the foreign DNA with the plasmid. So you have a cohesive piece of DNA here. Here's a picture kind of illustrating this. So we've got our restriction site. This particular restriction enzyme is only going to cut the sequence whenever it sees G, AA, T, C. Okay? And it's going to cut it in a ledge formation, or almost like a zigzag formation, leaving a sticky end. These sticky ends now provide the ledge for the DNA fragment to land on. So here's my foreign DNA. It's going to come in here, and it's going to bond at the sticky ends and really hydrogen bond to these locations because, they're attracted to each other in the first place. And then ligase comes down here and then covalently bonds at this location and at this location as well, and on the other sequence too. So what we've done there then is inserted our foreign DNA, and now we've got our recombinant piece of DNA here. So now we've got our recombinant DNA, but we've got to clone it now. So we've got to select a cloning vector, and actually here the cloning vector in this example would actually be the plasmid because it's carrying around the gene. So whatever is carrying your gene of interest is going to be your cloning vector, and the bacterial plasmid serves as a very good example of that. So I got a few examples in here just to kind of give you an idea of what we're looking at. Let's say our gene of interest was the beta globin gene in hummingbirds, and we wanted to clone that and isolate it later. Well, we would first find the hummingbird beta globin gene in the genomic DNA for the whole hummingbird, and we would isolate that. But then we'd have to do a critical step, and I want to highlight this step right here. We have to cut the DNA of the hummingbird and the DNA of the plasmid with the same restriction enzyme, so that when we stick them together, like on slide 10 here, they match. You can't just cut them with different restriction enzymes and expect them to match, obviously. Think of cutting a paper, like with crazy scissors, with wavy scissors, and then one with zigzag scissors. They're not going to fit together, okay? They're not going to look like they should fit together. So that's the same idea. So now we'll mix our fragments, mix in those sticky ends, and create our recombinant DNA. Once that's created, all we've got to do now is add that to a mixture of bacterial cells and then watch them grow. And actually, in most instances, you can grow them on certain types of agar. Now, agar is basically food for bacteria. So if you grow them on a special plate that just has the agar on it for the gene you're looking for the bacteria you're looking for, then only colonies of bacteria will show up if they have the gene of interest you're interested in. They won't just grow whether or not they got it, and you have this like giant field of bacteria where you're trying to sort out who's got what gene. So the special kinds of agar you can make will help you figure that out. And then the result is, well, great. Now we've got a lot of the gene of interest we're looking for. So that's really how you would create a piece of recombinant DNA and then clone it like that. So that's been going on for a while, and scientists have even been creating libraries of genes. So pretty cool. Here is that very thing right here. So we've got our genomic library that scientists are creating. Really what we're looking at here is taking the genome, not just genes, but the genome of certain organisms and copying those in bacterial cells. So we're taking every gene of a genome and putting each one of those into a plasmid. To give you an idea of what that would look like, here's my foreign genome. We're going to cut it with a particular restriction enzyme and then put every gene into a different bacterial cell, but then we'll grow them, and now every single well here in this plate will contain a whole host of bacteria with whatever gene we wanted to look at the moment. In an effort to speed things up, scientists have actually created a larger plasmid. So bacterial artificial chromosomes, or BAC chromosomes, are artificial, but they're larger, way larger. So now we can store way more genes in a single plasmid than just a regular bacterial plasmid, save us a little time, and then when we want a particular gene out of there, we just cut it out and study it at that point. So either way you go, you're storing genes in bacterial cells and creating libraries for these genes for later study. So we just talked about that. All right, so now we get to cDNA. And cDNA, or complementary DNA, comes about with the problem of RNA processing. Okay, we talked about in prokaryotes. Prokaryotes go from DNA to RNA to protein without any processing. Okay, there's no intron cutting or splicing going on. In eukaryotes, you have this RNA processing, all of this, right? All of this plus RNA processing. Okay? And if RNA processing is happening, we're cutting out the introns and we're splicing the exons back together. However, Bacterial cells cannot do that. So you cannot just copy a DNA sequence and put it into a bacterial cell and expect it to cut out the introns. So what are we going to do? Well, we're actually going to, in the lab, reverse this process and go all the way backwards. So we're going to have our mRNA strand that has already been processed. We're going to create DNA out of it. And that DNA is called complementary DNA because now we've made a DNA strand that essentially has no introns in it, which is great. So now bacterial cells can use that to make whatever protein we want to study. How's that done here? Well, we've got our DNA. This is the post-processing mRNA strand with our poly A tail and our cap here. We're going to make a piece of DNA off of that using an enzyme, a very special enzyme called reverse transcriptase. You don't see that in a lot of places, but there's a few organisms and viruses that have it. One very famous virus that has this is AIDS, or the human immunodeficiency virus, I should say. So anyways, we're going to create this DNA strand off of the mRNA. Then the mRNA strand is going to break up. We're going to make a complementary piece of DNA to go with it, and now we've got our cDNA with, miraculously, not miraculously, but you know what I mean, no introns. So great for us, great for studying this. All right, so now we've created our library, and we've got all these genes in well plates, and we want to see how different organisms compare with each other and how different genes compare with each other. And we may do that using a nucleic acid probe. Probes are great ways of finding particular genes of interest. So if you needed to do that, you could use a probe in the lab. Find genes of interest. Okay? The way you do that is nucleic acid hybridization, and this is just nucleic acid combination, or another way of thinking of it. So we're going to make a probe, which is a nucleic acid sequence, and we're going to bond that with whatever it matches up with. And if it does, then we know that it's containing that gene. So here would be an example. Maybe the gene of interest I'm looking for contains a particular unique code that is only, it doesn't have to be very long, but it's unique to this gene, or unique to what I'm looking for. So I would synthesize a complementary probe, and then all I would have to do then is insert the probe over my screen. So I'll take a kind of a screen or a nylon membrane off of these well plates, so I have a little bit of every gene, and I'd insert this probe on top of all those genes to see which gene had the particular sequence I was looking for. And it would light up when it found. So the radioactively labeled probe could light up under UV light, and then you'd have your gene of interest right here. And then you could extract that and use it. So you don't have to go through every single, gene and look, is this the gene that I'm looking for? No. You just highlight them all and see which one actually carries the gene. All right. So now we're on to expressing cloned genes. Typically, we talked about in protein synthesis how there are promoter factors, okay, or promoter / transcription factors that affect whether a gene gets copied or not. Sometimes bacterial cells will have a hard time getting that done because they are not used to expressing eukaryotic DNA. They may not have the expression factors needed to highlight the gene and get it transcribed in the first place. So some scientists will use an expression vector. The expression vector is typically yeast because the yeast cell is eukaryotic, yet it still has some basic prokaryotic properties as well. And in this instance, you can very easily express a eukaryotic gene. In cases where that's not working, some mammalian cells or insect cells can be used too, but expression vectors are important as well, because you can make all these copies of a particular gene, and then if you cannot express it in the form of a protein, or you cannot study the protein, then it's useless. So unless you're studying the gene, I guess. All right, so now we've got our complementary DNA, and we need to now study that, or we want to see how it operates in a different organism. We could take our recombinant DNA and place it into bacterial cells. We're actually going to do that in the lab, where we heat them up and cool them down, and in doing that, we'll create some holes, temporary holes in the bacteria, where we can insert the DNA. But that's not the way it's done for every single type of organism. You can do this in plants pretty easily if you were to use a type of formula or procedure called electroporation, where what we're going to do is electrically pulse the organism and create temporary holes, or minute little holes in the membrane where the DNA can get through. And once it's into the nucleus of that cell, then it can get copied and used. In creating certain GMOs, this is done in plants. So plants that are resistant to certain types of pests, so plants that are hardy winter plants, all of a sudden can have DNA inserted into them through this operation, and then grown into full-blown, full mature plants. So this can work as well. You can also, I guess, inject it to thin needles microscopically. That's still another way to go. But once the DNA gets in there, all it's got to have is an expression vector, and it can get copied just like any other gene here. Okay, this is actually really important. It goes back to what we were talking about in chapter 16 and 17. The fact that we can even do this is kind of remarkable. It's, we're taking, let's say, human genes, and we are inserting them into bacteria, and they are making proteins for us. Okay, or we study some of those proteins based on it. The fact that we can do that is pretty amazing. And when you think about that, and looking at the shared characteristics of DNA between bacterial cells and humans and other species, you can really see the underlying evolutionary ancestry of all those living species. We've all got the same DNA, leading us to the conclusion that all organisms have come from an organism with DNA, really. So, for example, here's a great example of this, and I think I got a video of this somewhere. I'll have to pull it out here for you guys in class. But the PAX6 gene, PAX6 gene is really kind of a I think that it's a gene that does not direct how an eye forms, but where an eye goes. And this is called a Hox gene. Okay, it really functions as a Hox gene, and Hox genes dictate where certain genes are expressed. So in humans, and actually in vertebrates, they did this with mice and flies. They were able to identify the PAX6 gene in a mouse and in an insect. However, a mouse has a regular camera vertebrate eye, and the insect has a compound eye. They were able to switch the two genes and grow or allow organisms to develop, and the organism developed perfectly fine even after switching the mouse gene for the fly gene. And all this leads you to the conclusion that organisms have really stemmed from ancestors that have shared these genes, and they're not completely unique to mice and flies. So it's pretty amazing that this is happening, or that this has something we can use to study gene expression with. Okay, so we're almost done here. We've got a few more slides to go, and then I think about 3 here or 4 to go, and then we'll be good to go for Friday. So the last thing I want to talk to you about in section 1 is how to amplify DNA. So DNA is actually really small. If you can imagine, you have a lot of it in every cell, but you have trillions of cells, so it's a pretty small molecule. in order to actually see it in a in any kind of experiment or study you're doing, you have to have a lot of DNA. Okay, so I'll just make a note of that here. In order to see DNA, you need a lot of it. And how are we going to get a lot of it? Well, one extremely simple way to do this is by doing a PCR reaction. We're actually going to do this in class 2. They take a very long time to run, but we actually have a machine, and there are many labs that have machines that will do this for you, where the heating and cooling process is robotic, and you don't actually need to sit there for hours and do this over and over again. So what we're going to do in a polymerase chain reaction is we're going to take the piece of DNA we want to look at, and we're going to make millions and millions of copies of this. And how we're going to do that is through a cycle. And the cycle pertains to just heating and cooling. And as we heat and we cool, we grow the population of DNA molecules exponentially. You may be asking and thinking to yourself, well, with heating and cooling, we're adjusting the temperature quite a bit. Don't enzymes fall apart when we do that? Well, there's actually a really interesting heat-stable enzyme called Taq polymerase that is quite expensive, and that enzyme is able to function in an environment where heating and cooling is consistently occurring all the time. So we use that one to get the job done. In PCR again, so we're going to take our one piece of DNA, and through heating it, we are going to denature the strands. So we're just going to kind of separate them. Once we separate them, we're going to anneal primers at the beginnings and end of the DNA strand, and then we are going to allow new nucleotides to join up using DNA polymerase, Taq polymerase, really, and then as we cool it down, those strands will anneal fully, and then we'll separate them again in the next cycle. So we keep heating and cooling, heating and cooling, heating and cooling, until we get enough of these molecules that we can actually see them. And you need a lot of them. So this typically takes about 6 to 8 hours to get done, and we'll run this throughout a whole day. So we'll have, multiple classes doing it at the same time, but we'll all do it together. Okay, so now on to what we'll do Friday. What we're going to do Friday is actually look at genes that are going to be inside of a gel kind of, bed, I guess I'll call it. And what the gel is going to be holding are different size pieces of DNA. What we're going to do in class is a little different from that, because we're going to save the DNA for a later lab, a week or 2 out. So we're just going to practice on Friday. But what we're going to do is the same technique as what we use in a week or 2. So what we do in gel electrophoresis is we just compare genes together. Okay, we're able to look at gene sequences and see which organisms or which individuals have similar genes to each other. So with gel electrophoresis, what we're doing is we are filtering the DNA based on size. Filter DNA based on size. And by doing this, you can see strands of DNA that are larger or smaller, and they will separate consistently throughout the gel, so you can compare different individuals or different species to each other. The molecules of DNA look like bands, and they separate based on the electrical charge. Since the DNA is negatively charged all the time, so DNA is negatively charged, all we got to do is place the DNA at the negative end of our electrified gel block, and the DNA will move towards the positive end, because the negative will attract to the positive. So here's a great illustration of what we're going to be doing. We've got our DNA, We've cut it up with restriction enzymes. We're going to use pipettes to insert that into wells at the top of the gel towards the negative end. And then when we turn on the power source, the smaller ends will move through the gel faster. Think of it like a giant sponge, okay? So when this is done, the smaller pieces of DNA will move towards the bottom, and the larger pieces will get stuck at the top and form a consistent banding pattern throughout the gel. So again, you'll be able to compare, well, did individual 1 have the same DNA as individual 2 when we cut it up with the same restriction enzymes? This technique is actually pretty interesting. It's used in crime scenes. It's used in, blood analysis. It's used in paternity testing. It's used in, comparing genes across species. There's I use this kind of DNA sorting technique in the research I did and for my undergraduate thesis, and looking at whether certain genes were in mosquito individuals or not across the same species in Florida, Alpha. So this technique can very easily be used to compare DNA strands. It doesn't take long to make and doesn't take long to run, which is why we're going to do it together, so give you guys some experience in the lab doing that. Okay, really, that's about it for today. I guess I'll finish up here with just ways in which you can analyze this stuff. Sorry, we'll be done here in a second. In analyzing these DNA sequences that I just mentioned, what we're really doing is doing restriction fragment analysis. So we're looking at the different size pieces of DNA and looking at how they relate to individuals. Again, we've just finished talking about this, but you can really use it to see where DNA strands form, or where DNA strands show up, and compare them between each other. So now we get to differences, right? So variation in DNA sequences is called polymorphisms, but looking at the changes in how a sequence can be cut. So you'll see in this picture back here, the DNA was cut at consistent spots, because it's the same spot for the same restriction enzyme. Not every individual has the same cuts. Some individuals have smaller cuts, whereas some individuals have larger cuts. And you can use this restriction fragment length polymorphism analysis to look at how different genes will show up on a gel electrophoresis read, and you can identify them as certain individuals if they have a particular gene. So the last slide here I'll show you for your example would be your hemoglobin gene. Okay, for beta globin, the allele cuts in, well, makes 3 pieces. Okay, so our beta globin gene in this instance would make this kind of, large fragment here. We'd have a 201 base pair segment here and a 175 base segment there. So we have 3 pieces of DNA after our DDE1 restriction enzyme jumped in and cut. Okay? Now we use the same restriction enzyme on someone with sickle cell anemia. It cuts in 2 locations, a large fragment and then a 276 base pair fragment. So when you run someone who has normal blood on a gel, their fragment analysis would look like this, where the smaller fragments would sink to the bottom, larger fragments would be at the top. So this would be their pattern. Someone with normal blood that they're compared to should have the same banding pattern. However, if they don't, there's a reason for that. And if this person had sickle cell anemia, you could identify the banding pattern in that way. That it has a large fragment and then a little larger fragment than these 2 down here. So that's how you could use this to identify certain genes. You could use to compare individuals. You could do a lot of stuff with that. So yeah, that'll be our intro for today in biotech. We'll finish some of this next week after we do the lab we're doing. I will see you on Friday, and if you have any questions, again, feel free to stop in and see me in the morning. Thank you, and have a good night.